Today we're going to be going over Chapter 9, the cytoskeleton and cell motility. Being motile means moving or having power to move spontaneously. Some background information is that the cytoskeleton is a network of filamentous structures. This includes the microtubules, microfilament, and intermediate filaments. Here we have a chart from the book that goes over the properties of microtubules, intermediate filaments, and actin filaments, and it compares them in dimensions, distribution, function, structure, subcellular distribution. This chart can be screenshotted and gone over because it has many details that can help you distinguish them. Now we're going to go over some of the main functions of the cytoskeleton. The cytoskeleton serves as a scaffold, providing structural support and maintaining cell shape. It serves as an internal framework to organize organelles within the cell. It also directs cellular locomotion and the movement of materials within the cell. Here we have how different structures are important to cytoskeletal functions. And all the structures labeled as 1 help with structure and support. Everything labeled with number 2 helps with intracellular transport. Everything labeled with 3 helps with contractility and motility. And everything labeled as 4 helps with spatial organization. Other important functions of the cytoskeleton include that it provides an anchoring site for mRNA, serves as a single transducer, and it is an essential component of the cell's division machinery.
Study of the cytoskeleton. The use of live cell fluorescence imaging can be used to locate fluorescently labeled target proteins. Molecular processes can be observed. Live cell imaging, used to reveal the locations of a protein present in very low concentrations. The use of in vitro single molecule assays. They make possible to detect the activity of an individual protein molecule in real time. Can be supplemented with atomic force microscopy to measure the mechanical properties of cytoskeletal elements, using video microscopy to follow activities of molecular motors. Microtubules, structure and composition. Microtubules are hollow cylindrical structures. The microtubule is a set of globular proteins arranged in longitudinal rows called protofilaments. Microtubules contain 13 protofilaments. Each protofilament is assembled from dimers of A and B tubulin subunits, assembled into tubules with plus and minus ends.
We're going to continue with microtubule-associated proteins, also known as MAPs or MAPs. MAPs comprise a heterogeneous group of proteins. MAPs attach to the surface of microtubules to increase their stability and promote their assembly. MAPs are also regulated by phosphorylation or specific amino acid residues. Microtubules as structural support and organizers. The distribution of microtubules determines the shape of the cell. Microtubules maintain their internal organization of cells. Microtubules function in axonal transport. Microtubules also play a role in axonal growth during embryogenesis. In plant cells, microtubules help maintain cell shape by influencing formation of cell wall. Microtubules are an agent of intracellular motility. They facilitate movement of vessels between compartments. Axonal transport. Movement of neurotransmitters across the cell. Movement away from the cell body and toward the cell body. Mediate tracks for a variety of motor proteins. There are motor proteins that traverse the microtubular cytoskeleton. Molecular motors convert energy from ATP into mechanical energy. Molecular motors move unidirectionally along their cytoskeletal tract in a stepwise manner. Three categories of molecular motors include the microtubule associated kinesins and dynesins, and the actin associated myosins.
Microtubules, kinesins. Kinesins, member of a superfamily called KLPs, kinesin-like proteins. A kinesin is a tetramer of two identical heavy chains and two identical light chains. Each kinesin includes a pair of globular heads connected to a rod-like stalk. Kinesins is a plus-ended directed microtubular motor based on its movement. They move along a single protofilament of a microtubule at a velocity proportional to the ATP concentration. Movement is processes. Motor protein moves along an individual microtubule for a long distance without falling off. KLPs move cargo towards the cell's plasma membrane.
Cytoplasmic dynein. Dynein is responsible for the movement of cilia and flagella. Cytoplasmic dynein is a huge protein with a globular force-generating head. It is a minus-ended directed microtubular motor, and it requires an adapter to interact with a membrane-bounded cargo. MTOCs. MTOCs are specialized structures for the nucleation of microtubules. The centrosomes are structures responsible for initiating microtubules in animal cells. They contain two barrel-shaped centrioles surrounded by PCM. Centrioles are usually found in pairs. Centrosomes are responsible for initiation and organization of the microtubular cytoskeleton. Microtubules determined in the PCM. Basal bodies and other MTOCs. Basal body structures where outer microtubules in a cilia and flagella are generated. Plant cells lack MTOCs and their microtubules are organized around the surface of the nucleus. Microtubule nuclearin. MTOCs control the number of microtubules, their polarity, and the number of protofilaments and their time and location of their assembly. The protein tubulin is found in all MTOCs and is critical for microtubule nucleation.
Microtubules. The dynamic properties of microtubules. There are four distinct arrays of microtubules in a dividing plant cell, widely distributed throughout the cortex, making a single transverse band in the form of a meiotic spindle as a pharmaplast, assisting in the formation of the cell wall of daughter cells. Newly formed microtubules branch at an angle of pre-existing microtubules. The changes in spatial organization of microtubules are a combination of two mechanisms: rearrangement of existing microtubules, disassembly of existing microtubules, and reassembly of new ones in different locations. Microtubules, the underlying basis of microtubule dynamics. Insight into factors that influence microtubule assembly and disassembly came from the studies in vitro. GTP is required for microtubule assembly. Hydrolysis of GTP leads to a replacement of bound GDP by new GTP to recharge the tubulin dimmer.
Intermediate filaments. Antiparallel associate each other. Heidi dynamics in vivo. Disappear before cell division and reappear in the daughter cells. Assembly and disassembly related to phosphorylation and dephosphorylation. Intermediate filament functions. They're found around the nucleus, terminated in the cytoplasmic plaques of desmosomes and hemidesmosomes. In nerve cells, they are referred to as neurofilaments. Here are some related diseases that we are going to talk about at the end. Microfilaments are good for migration, contraction, and motility. Wound healing, excellent growth, white blood cell migration, cytokinesis. G-actin transforms to F-actin filaments in the presence of ATP. Highly ordered form and tightly anchored bundle. Microfilament assembly and disassembly. In vitro, ATP actin monomers are the growing end. Barbed plus end is fast growing end, and pointed negative end is the slow growing end.
Cytochalasin from mold binds to plus end of the actin filament following depolymerization at the negative end. Phalloidin from a poisonous mushroom binds to intact filament and prevents their turnover. Latronsulin from a sponge binds to free monomers.
The molecular motor of actin filaments. Myosin is a superfamily generally divided into two groups, conventional type 2 and non-conventional. Move toward the positive end of the actin filament. Exit in mammalian cells, plants, non-muscle cells, protists, vertebrate, cardiac, and smooth muscle cells. Muscle contraction, the sliding filament model of muscle contraction, the composition and organization of thick and thin filaments, the energetics of filament sliding, excitation, contraction, coupling, and this will all be seen in the images in the next slide. Here we found a real-life example of muscle contraction in running, while sprinting, and while doing long distance. ATP is stored differently and released differently in both examples. Non-muscle motility. Contractile proteins are present in less ordered, more liable transient arrangements. They are restricted to a thin cortex just beneath the plasma membrane. Here's a list of actin-binding proteins: nucleating proteins, monomer sequestering proteins, end-blocking proteins, monomer polymerizing proteins, actin depolymerizing proteins, cross-linking proteins, filament-serving proteins, membrane-bounding proteins. Here are some examples of non-muscle motility and contraction. Actin polymerization as a force-generating mechanism, cell locomotion, axonal outgrowth, and changes in cell shape during embryonic development. Related diseases. Japanese quail-bearing mutation of neurofilament poses axons that are markedly thinner than normal. Mice carrying deletions in type 1 keratin are so sensitive to mechanical response that they can cause severe blistering of the skin or tongue. An inherited human disease, Desmond-related myopathy patients suggested from skeletal weakness, cardiac arrhythmias, and congestive heart failure.